We've already covered glycolysis and metabolism, but I want to focus a little bit more on what really happens in glycolysis in terms of the acid production, the hydrogen ions, the protons. So let's go through glycolysis again as a review, and then we're going to count and figure out where all the acid is coming from during metabolism to see what's really going on. So let's just do a quick thing here. Let's write the glycolysis down. We first start with glucose. Add a phosphate to there. Break that down. Aldehyde, 3-phosphate. Now we'll get our 3. Phosphoglycerate. Well, it's 1,3-phosphoglycerate actually. And then 3-phosphoglycerate. And then 2-phospho. Now we can form our phospholinopyruvate, or PEP. And then we can form pyruvate, which can go to form lactate, or goes into the mitochondria, forms acetyl-CoA. Okay, so let's just go through our glycolysis. So first, let's go through each of these steps. So first, glucose comes in, and we're going to make glucose 6-phosphate, right? So it's going to take ATP. We use ATP, and we get ADP. And then this will be the enzyme here, hexokinase. And now we have fructose 6-phosphate, fructose 1,6-bisphosphate. This will be another ATP. So we'll make ADP here. And this will be our PFK. And now let's go, of course, aldolase will break this. And now we'll get glyceraldehyde 3-phosphate. And we need have our NAD plus to make NADH. And this is done by the enzyme glyceraldehyde 3-phosphate dehydrogenase. We'll just write DH. And then our 1,3-bisphosphoglycerate, that's going to also give us an ATP. So this is our first substrate level phosphorylation. So ADP to ATP, and then rearrange that to 3-phosphoglycerate, and then 2-phosphoglycerate. That's a rearrangement. And then we get phospholinopyruvate. And so there's also a phosphate here. So we'll go ADP to ATP here. And this is by pyruvate kinase. Okay. All right, so we basically have our first, we could actually write some of these ones here. Phosphoglucose isomerase and some of these ones. Triose phosphate isomerase. And let's see, this one with enolase. We want to talk about this one as well. And of course, our phosphoglycerate mutase. Okay, now we have all these steps. Okay, so now let's talk about the acid, the hydrogen ions. Now, remember when we talk about hydrogen ions or an acid in general, we typically look at this equation right here. So we have HA, which is an acid, is going to be dissociated into A minus, which is the conjugate base, plus H plus. So this would be an acid, HA, because you get your conjugate base and then your acid, your hydrogen ion, your proton. That's your H plus. Now, so hydrogen ions then are the acid that we're talking about. Well, with glucose, we can actually, let's go ahead and count all the hydrogen ions, okay? So with glucose, we can put glucose right here. And the formula is C6H12O6. And when you metabolize your glucose molecule down to pyruvate, pyruvate is a 3-carbon molecule, and it contains 3 hydrogen ions, or just 3 hydrogens with it. So you end up getting pyruvate, and this contains, so it's C3H3O3, okay? And so in here, this is going to give you 3 hydrogen ions. So not hydrogen ions, there's 3 hydrogens. This accounts for 3 hydrogens. Now remember, there's 2 pyruvates, okay? See, this would be 2 pyruvates, because remember we broke this in half with aldolase. So there's going to be dihydroxyacetone phosphate, and then it's going to make glyceraldehyde 3-phosphate. So you would actually be getting, right, 2 of these. Okay, so there's 2 of every one of these ones here. All right, and then, of course, the 2 pyruvates we already wrote here, which means you get 2 lactates or 2 acetyl-CoAs. Now, so this accounts for the 3 hydrogens, but there's 2 of them, so times 2, right? So times 2 is equal to 6 hydrogens, all right? Well, this hydrogen, there's 12, right? So if we go 12 minus 6, well, that equals 6. So the question is, where are the other 6, the 6 hydrogen ions, right? Or the other 6 hydrogens, it's just a hydrogen ions. So the other 6 hydrogens, where are those ones at? And how many of those are H plus ions? So let's go through this reaction of glycolysis and figure out all the missing hydrogens. I'm going to use a different pen. And let's start with glucose. So the first reaction is glucose to glucose 6-phosphate. Now, what happens here is on the glucose molecule, you have basically this group of the carbon and then the hydrogen, okay, on the oxygen hydrogen. And you're going to switch that on this first carbon, so carbon, oxygen, and you're going to put a phosphate on there. So this hydrogen right here ends up being free into solution. Okay, so when this reaction happens, you end up creating 1 hydrogen ion. All right, so there's 1 hydrogen ion right there. Now let's go to the next one. So phosphoglucoisomerase, of course, nothing happens, it stays the same. Fructose 6-phosphate gets phosphorylated by PFK1 specifically, right? There's 2 PFKs, they're the ones, fructose 2,6-bisphosphate, which you want to remember because the fructose 2,6-bisphosphate allosterically regulates PFK1. Now here, though, we do the same thing. Look, this is fructose 6-phosphate. We're going to do the fructose 1,6-bisphosphate. So you're going to remove a hydrogen as well. So in this case, you're also going to be getting a hydrogen, all right? So we can put this one here. So we have H+. All right, now let's continue looking at these reactions. Glyceraldehyde 3-phosphate to 1,3-phosphoglycerate. There's no hydrogen ions that come off, or hydrogens in general, that come off of this molecule. So we don't include that. Okay. Now 1,3-bisphosphoglycerate, we're going to take the phosphate and we're going to put on ADP to ATP. There's no phosphate with that one, or hydrogen with that one. And now we have 3-phosphoglycerate to 2-phosphoglycerate. Then we have enolase. Now, enolase is an important one because you're going to convert 2-phosphoglycerate into this high-energy, rather relatively unstable molecule. And when it does that, you're going to create, it's a dehydration reaction. So what happens is you create water molecules. You create a water molecule when you do that. Now remember, there's 2 of these. So essentially you get 2 water molecules. And that means you get 2 hydrogens. But these are not hydrogen ions, right? That's not acid. These hydrogens are just accounted for in the water molecule. Not 2, but 4. You get 2 hydrogens per water, right? So you get 4 hydrogens in total. So now we can count them up, all right? So we can have, so here, let's count these up. So glucose has 12, all right? So glucose has 12 already, and now we're going to add in the 6 hydrogens from here, all right? So we get 6 hydrogens from pyruvate, all right? These are from pyruvate. Now we're going to get 4 hydrogens from water. And then you get 2 hydrogens, these ions, 1, 2, all right? So plus 2 hydrogen ions from water. And now you can add that up together, and 6 plus 4 is 10, 11, 12. And look at that. You get your 12 hydrogens, right? With 2 of these hydrogen ions contributing to the acidity, all right? There's 2 hydrogens directly from glucose that contribute to the acidity. So far we can see there's two hydrogen ions. So does that mean that glycolysis is acidic? Or what's happening? Well, let's continue going through because there's more reactions that we need to consider. We often, we have this reaction here with the glyceraldehyde 3-phosphate dehydrogenase. Again, this would actually be 2 here and 2 here. Now, remember when you often see in textbooks, it'll write NAD plus goes to NADH plus and H plus. Well, that H plus, like I said, doesn't come from the substrate at all. Where that H plus actually comes from the inorganic phosphate. So watch what happens. You have these PI, the inorganic phosphate that comes in, and that's why you have glyceraldehyde 3-phosphate, and then you're going to add a phosphate to each one of these molecules, so you have 1,3-bisphosphoglycerate. When it does that, you're going to end up coming out because it was attached to the phosphate. You end up getting 2 hydrogen ions, all right? Because this phosphate, just so you understand some of the way phosphate can be, the way how it exists. So you have this phosphate molecule, so PO4, and that's just a phosphate, but it has a negative charge to it, and it's going to accept hydrogen ions. So it's actually a buffer. So you can have it as, for example, if you have H3PO4, so it's like phosphoric acid, for example, right? Or you have H2PO4, or just HPO4. Okay? So phosphate can exist in all of these different forms. Phosphoric acid, this is not going to happen because it is extremely acidic, but it's going to be a mixture between these two, the diphosphate or just the monophosphate. And when your inorganic phosphate adds to this molecule, this hydrogen right here absolutely is going to come off. And this is why you get the hydrogen ions, all right? And so now we can go to glycolysis and we can actually add that. So we can say you get these 2 hydrogen ions here, Okay, these ones here are from glucose. And then you get plus 2 more hydrogen ions from the inorganic phosphate, all right? Now, those are acidifying, so we have 4 hydrogen ions, but there's more that's going on in these reactions as well when we start looking at substrate-level phosphorylation. Specifically, I want to draw your attention to this reaction right here with phospholinopyruvate to make pyruvate. This molecule, phospholinopyruvate, actually contains 2 hydrogens, and this reaction with pyruvate kinase is very favorable. And when it makes pyruvate, it actually takes a hydrogen ion to do that. So this reaction technically would include, so plus a hydrogen ion. And together, this makes the pyruvate molecule. Okay, so it's an actual, it actually is accepting a hydrogen during this reaction, all right? Now, we can then subtract some of these, okay? So now we can say, so minus, well, how many hydrogen ions is that? Remember, there's 2 of these molecules here, right? So actually, there's going to be 2 of these and 2 of these and be 2 of these. So we could subtract 2 hydrogen ions here, okay? And that's going to come from this reaction from pyruvate kinase. Okay, so now we've accepted 2 hydrogen ions over here. So now we subtract 2 hydrogen ions. But let's say we're going full-on anaerobic glycolysis, what we call, where we're going to make lactate. Well, we know what that equation is right here, right? Where we have pyruvate, and it's going to get converted to lactate. Well, what do we use? We use our NADH, and that regenerates our NAD+. Now, that's the hydride transfer, right? However, what's also going on, just as an exact same example as here, we're going to consume a hydrogen ion. So an H+ is going to be consumed in this reaction. And technically, there's going to be 2 hydrogen ions, right? Because there's going to be 2 lactate molecules, 2 pyruvate, and they're going to accept 2 hydrogen ions. So that's going to be another minus 2 hydrogen ions. And I want to explain this, actually, some of the chemistry so you can see why this makes sense. So let's go ahead, if we draw the different molecules, okay? So we're going to take, this is a 3-carbon molecule, CH3, and here. And if we want to go from pyruvate, I'll go from, pyruvate to lactate, right? So I'm going to draw here, see which one I want to do this. So you can see, right here I just want to show these, so far they look pretty similar, right? But here's where the difference is going to come in, is you notice with lactate, all right, we're going to say this is going to be lactate. I haven't finished drawing the molecule yet. With lactate, you're going to receive hydrogens, okay? A hydride transfer and a proton. And you're going to, it's going to look here. We can do OH right here and a hydrogen right here. First, we're going to write this one like this, okay? So, see, 1, 2, 3, 4, yeah. So here we have, if we count the hydrogens on this, we have 3 hydrogens right here. We have these 3 hydrogens right here. But look, there's 4, 5. So this one has 5 hydrogens. Where do these hydrogens come from? Well, this hydrogen right here was from the NADH transfer. All right, so if we draw, we have here NADH. And that's going to regenerate our NAD+. So this hydrogen, okay, right here, this hydrogen ends up becoming this hydrogen over here. Now, we have to add another hydrogen over here. Where did that hydrogen come from? That hydrogen was a hydrogen ion that came from solution. Okay, and that's That's where it came from. So this is the redox reaction of creating pyruvate to form lactate. You literally consume a hydrogen ion to make the lactate molecule. Now, this is still different than lactic acid, which I want to talk about in a little bit, but I just want to write what that would look like. Basically, if we would write, so lactic acid, we write the exact same structure, except you would add your hydrogen ion. So here we have our alcohol group here and your hydrogen. Now over here is where the difference is at. Instead of a negative charge on the carboxylate group, you would have this hydrogen. Okay, this is an acidic hydrogen. Okay, and this has what we call a pKa. I'll talk more about this, but it's a pKa of about 3.86. And essentially that means that when you have a pH of 3.86, then 50% of the molecules are in this form and the other 50% are in this form. 50% are in the acidic form, like lactic acid, 50% are in its conjugate base form, lactate, right? Acid form, conjugate base form. All right, that's all that it's saying. And it's pretty low, so 3.86. Okay, but we'll talk about that in just a second, but let's come back over here to glycolysis. We counted that all the 12 hydrogens are accounted for. We found that 2 of the hydrogens from glucose became acidic. We found that 2 hydrogen ions that contributed to the lowering the pH came from the inorganic phosphate right here. But then during the glycolysis reaction, we found that you are consuming 2 hydrogen ions from the pyruvate kinase reaction, and you're consuming 2 hydrogen ions from your lactate dehydrogenase reaction, right? Which is this, I'll just put LDH, right? For lactate dehydrogenase reaction. So what is the total then, right? What is the total hydrogen ions for the net acidity that you're getting? Well, this is a different equation here. So 2 plus 2 is 4, minus 2 is 2, minus 2 is 0. Look at that, so 0. So in this case, we would say glycolysis, based upon this stoichiometry, is 0, right? It's neutral, basically, right? So in this case only, this stoichiometry making, catabolizing glucose down to pyruvate is neutral. Now, then the question is, well then, what about all the acid? Where does that come from? And what about lactic acid, right? And lactate, because we hear a lot about lactic acidosis and lactate causing the burn and all these problems with lactate, right? Not so fast. There's a lot of really important benefits of lactate that I think it's really important that you understand. In fact, let's just talk about lactate molecule for just a minute, some of its benefits. When lactate is produced primarily in your type 2 muscle fibers, one of the first things that you're going to notice is that you can continue doing glycolysis. And this is the most important thing that I want you to understand is lactate production allows you to continue generating energy quickly. Think about what's happening here. We take glyceraldehyde 3-phosphate and we break it down to 1,3-phosphoglycerate. And that's an oxidation reaction. And if you use up all of your NAD+, because you only have a finite amount of those, okay? If you use up all your NAD+, and you convert everything to NADHs, you literally cannot do oxidation anymore. You're going to get a buildup of glyceraldehyde 3-phosphate, glycolysis is going to slow down, and you're going to stop making ATP. Glycolysis is one of the fast ways to get ATP other than, of course, your creatine phosphate system. So you have to regenerate NAD+ in order for you to continue doing glycolysis. So lactate production, right here, lactate production is absolutely essential for you to continue glycolysis so you can continue doing substrate level phosphorylation. And again, you're making 2 ATP. I'll just add that here for the record. 2 ATP, right? So you're getting a net of 2 ATP from glucose, or 3 if you're coming from glycogen. So this is a very important part. Just remember, lactate production allows you to continue doing glycolysis because you're regenerating NAD+. That's one reason why lactate production itself is very good. Okay, so I want to just let's see if we can list this out. Okay, here, NAD+ regeneration. Regeneration. Another big one is, notice this reaction, right? We're taking pyruvate and we're making lactate and we're consuming a hydrogen ion. If we're consuming a hydrogen ion, what's that going to do to the pH? It's going to raise the pH, right? It's a buffering effect. So lactate production, right, is it buffers, we can say, okay? It's a buffer because it consumes the hydrogen ion, okay? That's what we want to understand. Producing lactate literally consumes a hydrogen ion. Those are two key chemistry things that are going on. Now there's physiological benefits of the lactate molecule. Lactate is also a great energy source, okay? So it's a really good energy source. Think what you can do with that. The brain, for example, loves lactate. And when it's in the brain, it can actually lead to the increases in BDNF, which is the brain-derived neurotropic factor. Lactate can be metabolized really well by the heart is another one. The heart, you have type 1 muscle fibers. Type 1 fibers. And then, of course, it can go to the liver, and it goes through the Cori cycle, and you can then make more glucose or store it as glycogen, for example. If you think about how amazing lactate is, so your type 2 muscle fibers are rich in, they're rich in the glycolytic enzymes, and they don't have as many of the mitochondria and oxidative enzymes. So if you're doing a decent, exercise, your type 2 muscles could be engaged, and you could be making quite a bit of lactate. And as you make that lactate from your type 2 muscle fibers, that lactate will actually leave the muscle fiber, the type 2 muscle fiber, and it'll go into the type 1 muscle fiber. The type 1 muscle fiber can uptake that, and it can consume it as energy, convert it back to pyruvate, or there's even potential transporters within the mitochondria for the mitochondria to uptake lactate and convert that to pyruvate, and then go through the TCA cycle and get the energy through the electron transport chain. So this lactate molecule does so much because it helps to retard acidosis, it retards fatigue, it allows the energy to progress. So lactate becomes largely a hero in exercise. And so the question still is like, well, then why do we even talk about lactic acid? And the fact is, there really isn't hardly like any lactic acid at all, because what is a lactic acid? This is lactic acid when you have this hydrogen ion, right? So we can actually calculate how many lactic acid molecules there are at, say, a muscle pH is normally around 7. We can calculate how many lactic acid molecules are going to be there for every lactate molecule that you produce. And we can do this with what's called the Henderson Hasselbalch equation. All right, so this is just a simple equation. pH equals the pKa plus the log of your conjugate base concentration over HA. Okay. Now, the reason this equation is actually just a derivation from the Ka, from this equation. In fact, I'll just show you briefly how that happens. So if we will write the Ka of equilibrium, then we'd have this equation right here that we're referring to. Okay, so how do we do that? We do our products over reactants. So we have A minus, and we have H plus. We take a concentration of these. There's no coefficients here, so we don't need to raise it to the exponents over HA. And now we just have to solve this equation for H plus, because that's also what we want to do, right? So multiply both sides by HA, and we would do, we can do Ka times HA equals A minus times H plus. And now we can divide both sides by A minus, and I'll just put the H plus on the other side. So we can do H plus Okay, so we're going to this one here, showing the traces. H plus is equal to, we divide by A minus from both sides, right? A minus, those cancel out. And we would get Ka, and then we would have Ha over A minus. Concentration of that. All right, and now we can take the negative log of both sides. All right, so we go negative log of our H plus concentration is equal to, that'll take negative log of the other side, negative log of our Ka, and well, let's see, if we do HA, we can subtract those because the rule with logarithms, so we can do plus the log of A minus over HA. We switch those around, or we switch the sign. And now now remember, P, what does P represent, right? P is equal to the negative log. So now we can just put P in here. And what do we get? We get pH is equal to pKa, because it's negative log, pKa plus the log of A minus over HA. All right, so now we've just derived the Henderson-Hasselbalch equation. Now, our question initially was, well, at pH 7, how many lactic acid molecules are there? So we can rearrange this, and we can solve for A minus and HA. So to do this, we would take minus pKa over here. So we just do pH minus pKa is equal to the log of A minus over HA. And now let's get rid of the log term. So we could do 10 to the power of pH. Well, what pH are we saying, right? So this, we're saying at a pH of 7 in this case. So pH to the power of 7, and then minus the pKa, so minus 3.86. And that's going to equal A minus over HA. A minus over HA, which is the ratio we want to do, right? So 7.386 would be 10. So it would be 7.654. So 3.14. Oh, that's pi. Okay, so we have pi. And now we take 10 to the power of 3.14, and what do you get? Well, 10 times 10, 100 times 1,000, so you get like 1,380. So 1,380 lactate molecules for every, so per lactic acid molecule. Okay, there it is. So that means that you have to make, at a pH of 7, you'd have to make 1,380 lactate molecules to get even a single lactic acid molecule. And that's why we say there's like no lactic acid in the body. And of course, if you go to pH 7.4, right? So if you were to do, so this is pH 7, okay? This is pH is equal to 7. So at a pH of equal to 7.4, well, now it's going to be even higher, right? We do the exact same thing, and then so you end up getting like 3,467 molecules of lactate, per just one lactic acid molecule. Okay, so again, this is why we say there's like hardly any lactic acid in the blood. Now, what if we go more acidic? Okay, so let's go to a low pH. If we went to, say, a pH of around, the muscle pH really only go down to like 6.2, maybe 6.2 is pretty much as low as you're going to go. Now, if we do that, then you actually get about, so for every 200 molecules of lactate that you do, you'll make one, so you'll make one lactic acid molecule. All right. Now, let's think about this because this is actually kind of interesting. Now, at 6.2, yeah, now we are getting more lactic acid produced. But is this then making it acidic? No, it's the opposite. Because look at this. In order for you to convert your lactate, right? This is your lactate right here. To convert lactate to lactic acid, what is needed? What is needed? The hydrogen ion. Look at this. So plus H plus, right? That's what's needed. Because you got to make it right there, right? The hydrogen goes right there. This makes lactic acid. So actually, the lower the pH goes, the more lactic acid you make, and the more buffering you get. Does that make sense? So think about this again. Right here, you're only at lactate. If you lower the pH low enough, you start making lactic acid because lactate consumes yet another hydrogen ion to make the lactic acid molecule. And when the hydrogen ion is bound, it's no problem because it's not dissociated into the acid. It's not giving this free hydrogen ion into solution, right? So the production of both lactate is going to delay fatigue or delay acidosis because it's consuming a hydrogen ion. And the next step, the production of lactic acid, if that were to actually occur at a very low pHs and still it's a pretty high ratio, you're also consuming a hydrogen ion, right? So this really gets to the whole point that lactic acid itself from muscle metabolism is not directly the cause of acidosis as a proton donor, all right? So I think this helps to clear some of that up. However, it's a little bit more complicated than that because there is still a thing called lactic acidosis, where lactate can actually contribute to acidosis. And that's a different story because what we've talked about so far is this simple story, the simple story of acids being hydrogen ion donors. That's just a simple Bronsted acid, right? Or a Lewis acid, what we would call it. It's technically more correct to use physical chemistry to talk about this. And in this case, we need to talk about, we actually need to use mass charge balance equations. And this is to maintain the laws of electroneutrality. So just to understand a little bit of this, right? So we have electroneutrality. I'm just going to, that's that discussion so far. There's a principle in life called neutrality, electroneutrality, in physics and chemistry. So this is electroneutrality. And that is, you cannot have an imbalance in charges, in ions. I mean, yeah, you have like the cell membrane, so you have like a slightly negative here and positive over here, whatever, but they're right next to each other. So that's why you form this capacitance or like a voltage gradient, right? But they're balanced with each other. There's actually a balance between that membrane. So with electroneutrality, though, it's saying this. So it's really just this, that the sum of all the cations is going to equal the sum of all the anions. Okay? That is the laws of electroneutrality. And what we can do is, and these are the strong, all the anions, all the cations. And we can, there's a thing called the strong ion difference. And this is equal to your cations. Well, really, it's your strong cations. Your strong cations minus your strong anions. Because this is just the strong ones, right? So it's not going to include like your hydrogen ions, which is not necessarily it's not a strong ion because it's at equilibrium, or your bicarbonates, for example. If you do all that, you end up getting the strong ion difference, okay? Now, we won't go through all the calculations right now, but just to give an idea, we know that bicarbonate represents a very important buffer system, and bicarbonate is typically around 24 milliequivalents. I guess we want to write it here. Around 24 milliequivalents is the bicarbonate, okay? So that's your HCO3 minus. It's typically around 24 milliequivalents. And if we create lactate, then we have to maintain electroneutrality in order to obey the laws of physics, of chemistry. Okay, so think about what's going to happen then. We make a lactate molecule, and that's a negative charge. If you make a negative charge, what do you have to do so that's balanced? This has to be proportionally equal to a positively charged. Where does that positive charge come from? What's the only way to do that? It's going to come from water, and you're going to make a hydrogen ion. Remember we talked about this, the autoprotolysis of water? So H2O is going to go to H plus OH minus. So this reaction is always happening. So when you increase lactate production, you're going to have to increase H plus production. And the other idea is you can also decrease bicarbonate. So lactate production, so we can write, so lactate, what's going to have kind of two effects, right? So as lactate production goes up, that's going to increase your H plus concentration, and it's going to lead to a decrease in your bicarbonate levels. Because bicarbonate is also a negative ion, and so you want to make sure you have charge balance. Well, the bicarbonate is a big deal because that's our body's natural buffering system. Remember the equation where you have CO2 plus H2O, and you're going to form carbonic acid, HCH, form carbonic acid, H2CO3, H plus HCO3 minus. Okay, there's your bicarbonate. Here's our bicarbonate right here, right? As I said, there's like 24 milliequivalents. This is the body's main buffer system. So what happens if you increase lactate? So let's say you exercise, super high, right? You super maximal intensity exercise, and you increase lactate levels to around 20 millimolar, which because it's a one ion charge, that's going to be 20 milliequivalents. Okay, you increase this to say 20 milliequivalents. That means you have to keep the electroneutrality, and protons have to increase, bicarbonate is going to have to decrease accordingly. As you increase hydrogen ions, what's going to happen to the equation? Following Le Chatelier's principle, right? These are going to combine together to form more CO2, so you're going to actually lose bicarbonate. So whereas bicarbonate before was 24 milliequivalents, when you look at the phosphate buffers and everything else, it's going to decrease to around, let's say, 5 milliequivalents. Okay, now we can plug in 5 milliequivalents into the Henderson-Hasselbalch equation over here, and then we know what the pKa values are, and then we can solve for the pH. And at this level, your pH is going to be anywhere around like 7.2, for example. All right, we could do that equation. So this is why there is still a possible reason why lactate can result in acidosis. It's not because lactate is a proton donor, right? It's not. It's consuming a hydrogen ion. It still acts as it still can contribute to acidosis because of the laws of electroneutrality. This is part of physical chemistry, which we have to consider all of the charge and mass balance equations. And once we calculate that, we can figure out what the actual pH would be. So this becomes clinically relevant. As an example, you can have there's certain situations like with bacterial imbalances where bacteria is producing high amounts of D-lactate, which our bodies really don't metabolize the D form of lactate. And so that accumulates into the cells, it accumulates in the body, And that's going to be a problem because you get a whole bunch of lactate ions in the body, and you're not able to metabolize that very quickly. And what's that going to do? It's going to lower your bicarbonate levels, and you end up getting an increased hydrogen ion levels, and so you end up getting acidosis. Ketone bodies are another really good example. When you follow the proton production of ketones, and you do the biochemistry, most of your ketone productions are actually consuming hydrogen ions. And so it's like, well, then why do you get ketoacidosis? It's because you have high levels of ketone bodies in your blood, and you have to maintain the charge balance equations. And so your bicarbonate levels decrease and your hydrogen ion levels have to increase. And so this is why you get ketoacidosis, lactate acidosis. So this does happen quickly with your exercise, but it's not the primary. It happens. All this balances out very quickly. And actually, when you are, especially exercising really hard, then your CO2 levels are going to decrease a lot because you're hyperventilating, right? You're removing all the CO2, and that does help with this relationship as well. So you're able to decrease the amount of protons, hydrogen ions that are in the blood that would have been there otherwise. And the lactate production, it is another interesting point is, I mentioned though that lactate, like here, for example, we're getting up to 20 millimolar concentration. I want to add one other interesting thing about the benefit of lactate production. And that is, notice how I wrote that the lactate can be produced like in the type 2 muscle fibers, and that can be uptaken by the type 1 muscle fibers. When that happens, it actually goes through, so in your cell, all right, let's say your muscle cell right here, there's these transporters. These are monocarboxylate transporters. So we'll say MCT, like MCT1, for example. And these transporters can transport out monocarboxylates, such as lactate. Okay, so we have lactate right here, but it's a negative charge, and you can't take a negative charge out of this. And so it's actually balanced with a hydrogen ion. And so when it comes out, they come out together. So it's through a symport pathway. And hydrogen ions, the hydrogen ion concentration, right, inside of the cell, because you're exercising, it's quite high. It's much higher in the cell, because the muscle cell, the pH can be like 6.2, very, very acidic compared to the blood, 7.4, or in this case, like 7.2. But it's gonna shuttle out the lactate with the hydrogen ion. When you remove the hydrogen ion, when you take lactate out, you're also removing the hydrogen ion. So it's another way that removing lactate is actually going to help raise that muscle pH, because you're removing a hydrogen ion. So I just want to go through some of that, all those proton steps again. Think about what's happening when we create lactate. We consume a proton right here. So there's one proton we're consuming. If we end up making lactic acid, then we consume another proton, and then when we're shuttling lactate out of the muscle cell, we actually get rid of 3 hydrogen ions. So again, it really helps to illustrate why removing the lactate and production and removal of lactate helps to get rid of these hydrogen ions. And that happens right there in the muscle cells very quickly to help buffer everything. But again, we still have to worry about the laws of electroneutrality, right? So there's a lot of competing chemistry that's going on. Okay, so we've pretty much though explained that when you do metabolize lactate, all of this goes the other way, right? Like when you finally consume lactate, your reaction, you end up making your pyruvate molecules. Then that gets taken up to the TCA cycle, your NADH molecules. You end up getting to the mitochondria, goes through the proton, that makes a proton gradient. You make water. Oxygen is the number one reason, consumption of oxygen is the number one reason why we're able to consume hydrogen ions. Because remember the reaction, right? We have oxygen is what we're consuming. And when you combine that with 4 hydrogen ions, right? That's a lot of acid, okay? Hydrogen ions, plus, of course, your 4 electrons coming from NADH and FADH2. What do you make, right? These all together are going to form your water molecule. H2O2, so you have 2 water molecules. Okay, so this is why oxidative phosphorylation through the electron transport chain, it's not going to generate a whole bunch of acid because you're able to clear all the hydrogen ions that you're making, right? And so lactate, once you exercise at high intensity with your exercise, you make a lot of lactate. Lactate just goes to the liver, goes to the type 1 muscle fibers, goes everywhere, and everything just goes right back to balance, right? Your bicarbonate levels come back up, your NADH molecules get oxidized in the complex 1, your hydrogen ions get consumed right here with oxygen to make water, and water is good for you. We even drink this stuff. So this also explains why it takes some time to recover after a maximal sprint, or maximal exercise. So if you think about like a 400 meter, 800 meter, and you are going, all out, given all your gas, well, you literally made, think what you did to the chemistry, right? You depleted your creatine phosphate levels by like 80%, maybe a little bit higher. You made a bunch of lactate, and so all this equilibrium has shifted. Your bicarbonate levels are really low, and you have all this lactate out. Now you've got to get that lactate going back into the liver, the type 1 muscle fibers, and do all of this reoxidation and production of water. And that's why it takes 15 to 30 minutes basically to fully recover and get everything back to where it was before, where you have normal buffering systems. And so this is also why doing light exercise, getting your movement going, getting that blood flow is important after a hard, intense exercise, because you want to help circulate that lactate. From the blood, you want to get it back into the muscles, basically, your type 1 muscle fibers. You want to be able to oxidize that, but it's got to be low intensity, because we don't want to produce lactate. No, we want to clear lactate. And the best way to clear lactate is, well, the only way is with oxygen, right? So if you're doing high intensity exercise, you're just going to make more lactate. If you're just sitting around doing nothing, well, you need to consume, we could calculate this. We could calculate exactly how many molecules or liters of oxygen we have to consume to regenerate all of the NAD pluses, get rid of all the excess lactate. We can do that. And if you're just sitting there, like after you exercise, you're just going to lay down and you're dead, that's going to take so long for you to metabolize all of your lactate, regenerate all of the NADHs to go back to oxidize all your NADHs, right, and consume all your hydrogen ions to make your water. But if you're doing light exercise, well, now what's going to happen is you're going to be able to go you'll be able to clear all of that a lot faster, right? So that's the kind of the mechanism and science behind that. This is all this was fun, but it's actually not the primary reason why we even, where is the acid coming from, right? That's the real question. And acid, the hydrogen ions, the main hydrogen ions that we're getting is really coming from the hydrolysis of ATP. All right. And we can see that with glycolysis. And in fact, we can go through a number of very important steps when it comes to glycolysis, and we can see what happens with the hydrogen ion concentration, because this is only half the story. Now let's talk about the other half of the story, all right? Okay, so in order to understand some of the things we're going to talk about now, we need to talk about some of the substrates and products of the glycolysis pathway and what their specific pKa's are, because that's going to influence, of course, their hydrogen ion. Now, if we look at, so let's go through a number of different species, okay? So we'll go species. And of course, one of the first ones that we talk about is our ATP. Now, our ATP typically has a 4- charge on it, but it's almost always bound to magnesium. And that's why under the standard biochemical conditions, or G prime naught or G naught prime, we typically talk about having 1 millimolar of magnesium. So this, we actually would probably write MgATP, okay? And then we can look at the different pKa that it's going to have. So magnesium ATP is going to have a pKa of about 5 or so. And technically this would be what we call an apparent pKa, because it's not actually because of everything. It's all context dependent. What everything else is going on around there. Okay, so ATP. Then of course we break ATP down, and we would get ADP. And this one is actually a little bit higher. Now this is interesting because in free solution, your pKa they'd actually be reversed, where that of ATP would be a little bit higher than ADP, but that's not the case in here. And this is important for when it comes to some of these buffering effects. Okay, now we can break that again, and we have AMP, and that's not really associated with magnesium as much, just because the charge differences are going to be there. And this is actually a pretty good buffer. This is a pK of 6.21. And then remember some of the reactions we'll talk about, IMP, that can be broken down. And this is similar. It's almost a similar molecule because instead of adenine, you have ionosine. And so this is going to be around 6.2 or so as well. That's going to be another important buffer. There's just not a lot of it, right? When we do break down your ATP, that's when you form your inorganic phosphate. And this is one of the principal buffer systems, right? And this is around 6.82 and not really associated with magnesium as much. Now it can be, right? Especially complex with calcium. We talk about that when we're talking about fatigue mechanisms. But this phosphate, I kind of wrote it here. Let's just look what this phosphate looks like. So we have a phosphate, we have oxygen, 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 oxygen. And each one of these can have a different number of hydrogens, right? So these oxygens are negatively charged. Like I said, phosphoric acid, that will be a hydrogen on each one of these. And that's a very low pKa, right? It's just totally acidic. It's not going to be in solution. So you typically are going to have either here and here are your main ones you're going to have. And in fact, we could just we'll just do this exact same thing just so we can show the difference here. We'll give it to this one. So here, so when you have our inorganic phosphate at a pH of 6.82, if your pH, okay, if the pH equals the pKa, right? In this case, it's 6.82 for this inorganic phosphate. That means you would have 50% of it in this form and 50% in this form. We just talked about this before with the lactate and lactic acid. So this is why, so the best buffers by definition are those that they function the best when the pH is close to that buffer system, right? So of course a muscle pH is around 6.5 to 7 or so, and that's why having inorganic phosphate becomes a very, very good buffer, and that's going to come into play when we talk about the different reactions that are happening. All right, because this is 6.82, so it's right kind of in the sweet spot of being the perfect buffer. And it's going to exist in these two forms, right? This one doesn't have the hydrogen ion, this one does. Okay, and just as a note of chemistry, the reason why this one can donate its hydrogen ion is because this phosphate has a double bond right here, and so it's a delocalization of the electron. And same idea as I wrote over here, right? We have these double bond characters, so you can you basically delocalize the electron, and you can lose that hydrogen ion. You can, because this is going back and forth. A double bond can go here, can go here. It's just going back and forth. Pyruvate actually tautomerizes into an enolate form, but that's a different. This doesn't really matter. But this is why some of these are acids, whereas like this here, another acid, for example. Oh, here. This would not be an acid because you won't be able to stabilize this because carbon already has 4 bonds. You can't do 5 carbons. That's a carbocation. I mean, in transition states, you get some of that, but that's not going to make this one acidic. Okay, so just some chemistry with that real quick. Okay, now we have, these are our main species that we're involved in, that we're going to talk about. I guess we could also add phosphocreatine. That's going to be a big one too. So phosphocreatine. Now this one is an important buffer system, and the creatine itself is a Zwitter ion. It's not actually, this is not going to accept the, we just want to talk about the pK of the phosphocreatine. And this one is around 4.5. And another one we're going to come in contact with would be our ammonia. Okay, and this has a pK of around, I don't know, 9.8 or so. So it's very, very high. And actually, when it's in solution, it's going to be maybe not quite that high, probably closer to like 9.25 or so. This is going to be a very important one. So now let's look at some of our reactions. Let's start with glycolysis, and then we can see what the effect of each reaction is going to happen on the pH, okay? So let's look at our first reactions over here. So if we look at various reactions, okay, so we have our reactions, and let's take a look at our, what we could do is we could go out and put in each effect. So, for example, we'd have some that are direct H plus ions, and then you're going to have like a nucleotide effect, or for the phosphate. Okay, they have nucleotide. I'm thinking your phosphates. Those have different buffering effects, right? And then you'll have, depending on the substrate itself, we can just say some other types of effects if they apply. And then we'll have the total effect, okay, the result. We can just put the effect, I guess. And we can now look and see which each of the reactions are gonna happen, all right? Okay, so when let's look at all the reactions, right? So our very first reactions, we have hexokinase. Okay, so we go hexokinase, and does that give us a direct hydrogen ion? Right, it does. So we have 1 H plus right here. Now we look at the nucleotide and the phosphates. Now, what's happening right here? We are in the reaction, we're taking ATP to ADP. So we are consuming ATP. And when you do that reaction, if you look at the pKa's, notice ATP, as I said, has a slightly lower apparent pK than ADP does. So actually, ADP is a slightly, when this reaction happens, you're actually going to consume a little bit of hydrogen ion. It's a very, very small amount. Okay, so you could say it's like negative 0.015. It's a very small amount. And now you could also take a look at the molecule itself. So glucose 6-phosphate, right? Does that have any buffering effect, the actual carbon substrate? And it does, but you're going to see throughout these, every substrate and carbon has a slightly different buffering effect. They have different pKs, different apparent pKa's, and we would consider all of those. But I did all of that, and I realized it doesn't actually matter, because in the end, you start with glucose, which has no acid hydrogens, and you end with pyruvate, which is totally in its pyruvate carboxylate form. So there are no hydrogen ions associated with that. So all of those end up canceling out anyway. So it actually doesn't really matter. So we can just start with, we can skip that, and we'll just start with the main reactions that happen with our direct contribution and then our nucleotide effect. So of course, if we add this up, then you'll get the total effect of 0.871 or so. All right, so pretty much a full proton. All right, now let's go to the next reaction. What do we see? So we get PFK1 specifically, right? We're going to take ATP and so it's the exact same thing that we're happening. So we go PFK1. We are going to contribute 1 hydrogen ion for sure. We're having that proton, the changes in the phosphate species, right? So we are going to buffer a little bit better with ADP than with ATP. And then you're going to get the same 0.871. So still pretty much a full proton, right? Okay, so now we continue looking at this, and what do we see? What are the other sorts of protons? This one here, the glyceraldehyde 3-phosphate dehydrogenase, right? We do get protons from there. So we can just write gap DH, and we get 2 hydrogen ions from that step. Well, actually, we can write this differently because technically this does not come, these hydrogen ions, right? They actually don't come from the glucose molecule. Technically, they only come from this guy right here, from the actual, the change of the inorganic phosphate, because initially you had this guy right here, and you switch all of them to this guy right here. And so you end up losing that hydrogen ion. And that's the hydrogen ions that we're talking about. So this right here would contribute essentially +2, because you get the entire, there's going to be 2 of those for each one of the reactions, right? Now, there are some shifts as well from like the glycerol from the phosphate molecule, but we don't have to necessarily include those ones as well. However, we also need to include some other aspects, because you're actually changing a number of species, because this is just one of them. But remember, if at a pH of, let's say we're at a pH of 7, okay? This whole thing is technically going to be at a pH of 7. And that means that you're going to have a certain percentage of the molecules that are in this and a certain percentage that are molecules that are here, right? Of course, at a pH of 7, most of your phosphates are going to be in this form right here. But you're going to have a pretty decent amount, like 40% or something, that are going to be at this one right here. So you actually, if this guy right here attacks, in this nucleophilic attack, sorry, where is that? Yeah, this one. If you do a nucleophilic attack with this inorganic phosphate that contains 2 hydrogen ions, then you're going to get both of these hydrogen ions, 2 of them, right? You can imagine if both of these ones attacked, this would actually give you 4 hydrogen ions. And so you actually have to calculate what is the pH, and then what is based upon the pKa of both of these, that's going to tell you what the true hydrogen ion is. So we're just using 2 because we're saying we know for sure 2 is going to happen, but that's only if all of them are in this state. But like I said, some of them could be in this state. So at a pH 7, if you did the Henderson-Hasselbalch equation, you would find that it's going to be more than just 2. It'd be closer to 3. So it's actually like 2.796, if I remember right. So it's pretty high. Okay, that's actually what you're really getting at a pH of 7. Of course, that's going to change. 796. That's going to change if you are different pHs, right? If you're more acidic, then this can be even more, right? Because you're going to get more of those protonated. So your total effect then is 2.796. Okay, so that's a major reaction here, the glyceraldehyde 3-phosphate dehydrogenase reaction. All right, now we go down and we find the next one. What's the next one that's going to be important? Well, you can look at the changes of like the phosphoglycerate mutase. You do change the structure, and when you change the structure of these molecules, you do end up changing the pKa's a little bit, and they're accepting hydrogen ions a little bit, they're releasing some a little bit. But again, as I said, they all wash out in the end, so actually it really doesn't matter. So the next thing we have to just look at is jump right to pyruvate kinase over here, because we're accepting that hydrogen ion. That's going to be the next important one. Okay, so we have pK. Now, in this case, though, are we consuming a hydrogen ion, or are we producing a hydrogen ion? We're consuming one, right? And there's going to be 2 of them. So in this case, it's going to be 2 hydrogen ions. And this is going to be by definition because we're literally putting a hydrogen ion onto the actual molecule. So we don't have to worry about all the buffering things with that actual reaction. But there is ADP that's getting converted to ATP, right? Remember that ADP is able to buffer more hydrogen ions than ATP is. So that also contributes a little bit to the actual acidity. Not very much, right? Just like we talked about earlier, this is going to do a little bit. And so because there's two of these, then we could just add these ones together, right? And it's going to contribute to about 0.03. So not very much at all, but it's still, there's something there. And so if you put these together, then you would get, right, so 1.97, for example, okay? So again, you're getting most of them is, you're getting rid of that hydrogen ion, so that's ideal. Okay, so now we've done all the reactions. Of course, we can include our lactate dehydrogenase reaction. Okay, so lactate dehydrogenase reaction. And is that consuming or producing hydrogen ions? Well, in this reaction that we're talking about, we're taking pyruvate to lactate. We are going to be consuming our hydrogen ions. Well, consuming, so that actually we're gonna take it away. We're gonna consume these, so -2 hydrogen ions. And there's no nucleotide effects in this reaction, right? It's just NADH. And so it's just going to be 2, right? And now we could actually just take and total this all up. So what is the total of all of these together, right? So here we have 1, 2, and then 1, 2 positive, -2, -2. And what do you get, right? -2 hydrogen ions. So when we include lactate, we're actually getting a net of -2. So glycolysis with the acid production, so far it looks like it's literally alkalizing, right? That's a crazy thing. The glycolysis itself can be alkalizing. However, it does get complicated because when we add up all of these, right? Especially this 2.796. When we do that one, it's going to be like 2.825 or so, okay? So now when we add all of this up together, right, the 2's cancel out, and so we end up getting like 0.82 net hydrogen ions. So in the end, glycolysis ends up becoming slightly acidic, even if we include the lactate dehydrogenase step. And that's primarily because of the breakdown of glyceraldehyde 3-phosphate. Because you add these, okay, it's not the breakdown of glyceraldehyde 3-phosphate, it's the addition of these inorganic phosphate molecules because you have these two species right here. And again, this is at pH 7. So at a pH 7, you're contributing about 0.82 hydrogens, 0.82 hydrogen ions to solution doing glycolysis. Now, that's not all the reactions, right? There's other reactions. Let's look at the other reactions that are happening along the same time that we can include in this. So the first important reaction here is, I just want to do our ATPase reaction, okay? So our ATPase. And this is when you simply just take ATP plus H2O and you break it down and you get H plus ADP, right? That's the most straightforward reaction there is. And when we do that reaction, how many hydrogen ions do we end up getting? With this reaction, you get +1 for sure, right? It's right there. It's that simple. However, you took ATP and you converted this to ADP. And remember that ADP is able to buffer a little bit better than ATP is. And so this is going to be slightly disbalanced with the production of ADP. You can actually buffer a little bit more, all right? And what is that number? So we come over here and we make our ADP molecule, and this ends up being around negative 0.413. All right. And so when you put these together, right, you have one, you're like pretty much at a half. So you end up getting like 0.587. So this buffer here with ADP is actually a big deal. It's very important, right? Okay, let's go through some of these reactions. What other reactions do you remember? We have our creatine kinase reaction. Okay, so our CK. And what does that reaction do? We take our ADP plus H plus, and we make ATP plus our creatine. Again, look at this, H plus. So we have a direct hydrogen ion right here. So there's plus 1 H plus, okay? And now, but we converted ATP to ADP to ATP. What effect is that going to have? Well, again, because of the buffering situation, you're actually going to add a little bit, right? Same thing over here. So here you add like plus 0.015, okay? And so it's going to subtract a little bit from the from this one. So 0.9 0.987. Okay, so still almost a full hydrogen ion, but I guess something to consider. Okay, what's another important reaction? How about this reaction where you get 2 ADP and you're going to put those together, and what are you going to get? ATP plus AMP. Remember that the enzyme is called the adenylate kinase? What are the effects on this case? We can start to predict what's probably going to happen, right? There's no direct hydrogen ions that are used here. We do change this ratio of the ADPs to ATP, but the AMP, when we look over here, the AMP is actually is a pretty good buffer as well. And so the buffering effects, they overcome the impaired or the lower buffering effect of ATP compared to ADP. So the net effect ends up being a slightly better, right? So around 0.1 or so. So here you get 0.1. Okay. Good. We have, I think I got our rows a little bit off, but yeah, this is all. Okay, 0.1. Okay, so that's where our effect here is, and then we don't need to worry about the other, and so our full effect is just, right, 0.1. All right, so that's our net effect. So it's slightly alkalizing, but it's pretty much meaningless, right? And then we made our AMP. Do you remember what we do with our AMP molecule? Remember what that reaction is? We can take our AMP and we can convert that to IMP plus ammonia. And it does this with AMP deaminase, right? Put deaminase. And AMP and IMP, remember, these ones are pretty much the same molecule, right? So you're not going to make too much of a change with just adding this one. So there's hardly any change, but one critical point is that we are having this ammonia, right, that was formed. Now this ammonia is really, ammonia is toxic to the cells. It'll totally destroy the cells. So, and ammonia has a very high pK, right, like 9.25 in this scenario. So what's going to happen? I'm just going to write this reaction here. So we would take ammonia plus a hydrogen ion, and it's going to form ammonium, okay? And this reaction will certainly happen very quickly and to completion because the pKa is so high. So this is why this reaction becomes very alkalizing. So we can just say you're getting, well, it's going to be minus. You're getting a full hydrogen ion that's being consumed by this reaction. Very important. In fact, this AMP deaminase, not the reaction itself, but because you form ammonia, and ammonia is going to consume a hydrogen ion, absolutely, to make ammonium. That becomes a very important proton-consuming reaction. So when you're hitting the gym, for example, and you're lifting like really heavy weights, and you are breaking down that ATP to ADP and AMP very quickly, the AMP goes to AMP deaminase, and you're able to buffer the hydrogen ions that you're producing because you're forming ammonia, and then ammonium gets produced immediately after. So it's a very important reaction, actually. But it's not great because ammonia is toxic for you, and then ammonium will get tagged onto different amino acids like glutamate to form glutamine, and then that's the carrier, and it goes through the urea cycle, and you can metabolize everything, right? So there's a way to handle all of this, the urea cycle. Okay, so that's an important reaction. Now there's another, and I guess we can think about other important reactions as well. Well, these ones are our main ones, but if we think about what's going to happen in reverse, right? So we talked about already our creatine kinase reaction, but it's happening this way. It's happening forward, but we could say what's going to happen if it goes in reverse, and like what happens like in the mitochondria. So in this case, you would have ADP plus your creatine. Oh, sorry, ATP, because we want to go reverse. So you have ATP, and what are you going to form? Your creatine phosphate plus ADP. But also, when this happens, you form your hydrogen ion, right? It's just essentially it is the reverse reaction of that one. So that ends up being more acidifying in this case, which is okay when you do it in the mitochondria because you're already consuming a lot of the hydrogen ions to make your metabolic water over here, so it doesn't lead to an increased acidity. You're able to handle that, no problem, with the enzyme creatine kinase, more in the mitochondria. Okay, so you end up getting +1 in this case, right? Okay, so now we've gone through all the core reactions, and let's just put a refresher on this really quickly. When we do glycolysis, we found that only 2 hydrogen ions are contributing to the acidity. But during glycolysis, with your reaction on pyruvate kinase, those hydrogen ions are being consumed in the phospholipyruvate to make pyruvate, and so you end up getting a -2 hydrogen ions. So that portion becomes quite alkalizing in and of itself. But you still are making, and then when you include lactate production, you consume those other additional hydrogen ions. So glycolysis by itself is largely neutral from the carbons. However, it still becomes acidic, and that's because of this glyceraldehyde 3-phosphate dehydrogenase step. Not the reaction itself or the breakdown, but because of the inorganic phosphate, you're producing close to 3 hydrogen ions, not just 2, because of this scenario right here. And that's why I wrote it up right here, right? pH 7, 2.796. And that's why when you add everything up together, glycolysis ends up becoming 0.82 as a net proton delivering, right? And of course, this is going to change with the pH. And then when you are exercising hard, the very first thing that's going to happen is you're going to consume ATP. You make hydrolysis, but for every ATP hydrolysis reaction that you're doing, look at this. You do produce a hydrogen ion, but you're also producing ADP, and so you're able to consume some of those hydrogen ions as well. And plus I don't have the phosphate. Some of the plus ADP plus your inorganic phosphate, right? And so you're able to consume some of a portion of that hydrogen ion. And so you're only making like half of a hydrogen ion, again, at this pH. And so, but because you're doing so much ATP hydrolysis, obviously this wins out and you end up acidifying. But keeping it just with our ATP producing pathways, our synthesis pathways, our creatine kinase reaction, that's consuming a hydrogen ion. And then our AMP deaminase, that consumes a hydrogen ion. So even though you're getting a net of 0.82, when you add that, when you add this one right here to what you do with creatine kinase, right, those are going to cancel out. And then you have this one right here, that's going to be -1, and then you have this, only about a half of an ATP right here. You can see that these reactions, in the end, all sort of neutralize each other, right? And that's going to be important, again, to buffer those hydrogen ions. Now, you're still producing a lot of lactate, and then, of course, this portion of the electroneutrality starts to play a role, and you start lowering bicarbonate levels, and so your acidity levels start to happen from that mechanism. But again, most of the acid that is being produced is truly just from the hydrolysis of ATP, from your myosin ATPase and your circuit pumps, sodium potassium ATPases, right? There's a lot of pumps and ATP currency being used very quickly, and you are breaking it down, which is going to contribute to the hydrogen ions. But you do get some inorganic phosphate that's going to be produced, which is going to help to buffer some of that, but not as much as the actual hydrolysis of ATP contributing to that hydrogen ion. Okay, so now that's the effect with glycolysis. Now we can follow a similar pathways as we go through the oxidation of fatty acids in the mitochondria, and we can see, okay, is that reaction going to be more net acidifying, neutral, or alkalizing? All right, let's take a look at that. Okay, but before we do that, I just remembered with lactate, there's one other really cool thing about lactate. It's kind of recent, more looking at it, but we've talked about how lactate serves as a metabokine, right? It's a metabokine. And it turns out that you can modify histones, so histone modification, through this process of lactylation, where you actually are adding lactate molecules to the histone, and that's going to have epigenetic changes. So it's another actual key benefit of lactate. One of the reasons why maybe exercise is good for you, because you're changing things at the epigenetic level. So again, lactate has lots of benefits, precursor to gluconeogenesis, and anyways, we've talked about things already, but just a cool thing about epigenetics aspect of lactate also. Okay. All right, so now let's go to mitochondrial respiration. Just a quick overview, and then we can talk about what the net effect is going to be in terms of the acid metabolism, right? So again, let's just draw a mitochondria. Okay, so before we go into the hydrogen ion situation with oxidative phosphorylation, let's just do some quick review. So here we have, looks like a rainbow. It's actually the mitochondria. Of course, they do not look like this. Normally have like all the folds and everything in the mitochondria, but I'm just trying to show what we have here, right? So here we have the outer mitochondrial membrane, and this one is the inner mitochondrial membrane. And we remember that we made our NADH molecules, right, in the glycolysis. And these NADH molecules, as I mentioned before, they actually cannot, they cannot permeate this inner mitochondrial membrane, okay? So this membrane is impermeable to them. And so they have to go through different mechanisms in order to enter into the inner mitochondrial membrane. And that's true with, like we talked about with beta oxidation, with fatty acids. Remember, we would take our fatty acid, for example, and we would have to add a CoA, right? And this CoA required an ATP in order to do that. And let's see. Yeah, so it required an ATP, actually it would be an ATP, but eventually what you're really doing is you're breaking it down to inorganic phosphates, right, where you get your pyrophosphate. So it's equivalent to basically 2 ATP. This is going to come in handy later. But once you make you put your CoA on there, then you can take the fatty acids and you go through this shuttle, right? This is the carnitine shuttle, so CPT1, for example, and you have your carnitine molecule that's in here, carnitine, and you're able to shuttle, you switch this out, right? The CoA and the carnitine, they switch, and then the fatty acid is able to come in right here, and then you have the carnitine ends up coming over here, and finally the fatty acid comes out. You put another acetyl-CoA on there, and now you're able to do beta oxidation, right? And then the carnitine just switches back and forth between here. So, and remember that all the fatty acids that are, basically larger than 12 carbons, they have to go through the carnitine pymidyl transferase shuttle, right? So here we have CPT2, for example. And this requires carnitine. And the idea is, of course, you take carnitine, then you can help oxidize more fatty acids, but that is not the rate-limiting step of fatty acid oxidation, right? All the other processes are the rate-limiting step. Once you get into the matrix of the mitochondria, then you can undergo beta oxidation of the fatty acids, and you basically go through each of these steps we talked about before, and you end up getting your FADH2s and NADH2s, or just NADHs, and then you can oxidize them in the mitochondria. I will just add one point. So when you go through the beta oxidation, let's say you have this is a 16 carbon molecule. Okay, so C16H32O2. You go through beta oxidation. You know how many cuts that is, so you end up getting 7 FADH2, and you get 7 NADHs. Now, this FADH2 it is different than the TCA cycle FADH2, which is over here. There's a different step in order to do that, because the FADH2 is a membrane bound, so it's a different one. We'll come back to that in a little bit. Okay, we want to talk about, let's first just start with glucose. Now we get pyruvate. Now pyruvate is going to come, as we talked about before, pyruvate, there is a shuttle where pyruvate is able to come through. So we have pyruvate, and also comes with a hydrogen ion, Another way to take up hydrogen ions from the cytosol. That's a cool thing. We come into the matrix, and we get our hydrogen ion over here, and we get your pyruvate. And then pyruvate goes through the PDH complex, and ends up getting converted to acetyl-CoA. Acid. All right, now acetyl-CoA, and then it's going to combine with citrate or oxaloacetate to form citrate, and you do the entire TCA cycle. All of that does not require any oxygen. Okay, when you do that, you end up creating your NADHs and your FADH2s. Okay, so once we do the full cycle of here, every time we make a turn, the entire turn, what do we end up getting, right? Well, pyruvate turns into 2-carbon acetyl-CoA, that goes to oxaloacetate. We can count each one of the steps, and so how many FADH2s and NADH2s do we get? All right, so NADHs and FADH2s. So you should know how many we're going to get at this point. So if we include everything we get, like from glycolysis and the TCA cycle, right, we're going to get 10 NADHs, and then we'll get 2 FADH2s for every glucose molecule, right? Because remember, this is going to go through the cycle two times, because you have 2 pyruvates that are entering into the cycle. Now, what do we do with these electron carriers? This is where we have the electron transport chain, right? So remember, we have complex 1, we have complex 2, we have complex 3, we have complex 4, and we have complex 5, or really this is the ATP synthase, right? This is a little nanomotor that we talked about. And here we're going to take our ADP, make ATP, right, plus our inorganic phosphate. And we remember that every time when we do ATP production, or we're driving these electrons, complex 4 right here, this is where oxygen is being consumed at, right? So we can take our oxygen molecule and we're going to combine that with 4 hydrogen ions, and it's going to get 4 total electrons, so plus 4 electrons, and that's going to make our water, H2O, right? And remember that we get those hydrogen ions are going to be pumped through right here, right? So you get H plus ions. So you have a lot of H plus ions that are being pumped into right here. And this is complex 1, so this is where you take your NADH, and you get oxidized to NAD+. Here's your FADH2, you get FAD. And remember, this is the same succinate dehydrogenase complex that's in the TCA cycle. And then we have this complex here, which, okay, so you have the electrons. As these get pumped, you have your CoQ10 and basically, right, your ubiquinol. And it's this electron carrier right here is going to carry your electrons to complex 3. And when you do that, you also get hydrogen ions, you get proton pumping, right? And then finally, you get complex 4, where we say you consume 2 hydrogen ions with half of oxygen water. And then you make your hydrogen ion gradient really high right here, and then your hydrogen ions are able to come through this little nanomotor, and that's when you're able to make your ATP. Okay? Basically, it's just putting ADP and PI together. It's just pushing them tighter and tighter until you make your ATP molecule. Okay, we've already talked about all of that. And we talked about how all these NADHs have with them 2 electrons. So if you drop electrons right here, then you're going to have 4 pumping, and then these 4 pumping, and so that's going to be 8, and then you get just 2 hydrogen ions right here because you're getting consumed by oxygen, and so that's 6, and so this is why each NADH molecule is worth 2.5 ATP, right? So NADH is worth 2.5 ATP, whereas if you use the FADH2 right here, you skip this proton pumping action. And so in this case, it's only going to be worth 1.5 ATP. And so when we look at the glucose metabolism, when we get pyruvate, we have these NADHs over here, but the 2 NADHs over here that were produced in the cytosol, it depends on the cell. So like muscle cells, actually, they don't go through this malate aspartate shuttle that we talked about earlier. Instead, they go through the glyceraldehyde dehydrogenase, or the glycerol 3-phosphate dehydrogenase shuttle mechanism. And they're going to drop their electrons off over here to complex 2 with your NADH, NAD+. So they end up skipping those electrons. And so we calculated out that a 1 molecule of glucose is going to produce either 30 or 32 ATP, right? When you calculate the 2 GTP from the TCA cycle, which is equivalent to ATP, and then the 2 ATP from glycolysis. So you add these up, you get 30 or 32 ATP. And we talked about how you're consuming electrons and hydrogen ions right here. Now, earlier in glycolysis, we counted and followed every one of those hydrogen ions. Well, we can do the same thing right here. We're pumping for hydrogen ions. We have pyruvate, 2 pyruvate molecules that entered, and there was 6 hydrogen ions in total, right, that came in. So there were 6 hydrogen ions from pyruvate that came into the matrix. And yet, in order when NADH comes and we oxidize this is getting 4, 8, 9, 10. So one NADH molecule that gets oxidized all the way by molecular hydrogen, it ends up consuming or basically using or pumping a total of 10 hydrogen ions. But I guess you can look specifically though at the stoichiometry, right? Is there's 2 hydrogen ions that get consumed for half a water molecule. So a full water molecule, full oxygen molecule. So full oxygen molecule, O2, is going to use 4 hydrogen ions, right? So 4 hydrogen ions and 1 oxygen molecule is going to give you 1 water molecule. So you get 6 hydrogen ions from the 2 pyruvates, and you are consuming 4 hydrogen ions right when you use a full oxygen molecule. Now, obviously, you're making a lot more NADHs and FADH2s. And so your net effect is you're going to be consuming more hydrogen ions than you brought in. You only brought in 6 hydrogen ions. Remember, all the pyruvate molecule, because this gets decarboxylated, right? So you leave as CO2. And so now you just have a 2-carbon molecule that contains 3 hydrogen ions, right? So it'd be C2H3O2, right? And this whole thing gets oxidized to just make your CO2. So you get 2 CO2 molecules. That's it. And yeah, so the hydrogen ions get put into this matrix and they end up as water, and going through the ionic product of water, and the hydrogen ions get pumped out and consume with the oxygen, and then you make water. So the net effect ends up being a net consumption of hydrogen ions. So this is why oxidative phosphorylation ends up becoming quite alkalizing. In fact, we can write this if we do a little. Okay, so we'll write glucose up here. So we have C6H12O6. And we want to do the complete oxidation of glucose. And we're going to combine that with oxygen. And you're going to get 6 CO2s plus H2O. Well, we know how many oxygens are needed in order to completely combust oxygen. We can actually count all of them. We did that before, and we found there's 24 electrons in total, right? Because you have 10 NADHs, 2 FADH2s, and so that's 12. And so you can do 12 of those electron carriers, right? So 12 NADHs and FADH2s. And that's going to give you 24 electrons. And then if you take 24 electrons, and we know that the stoichiometry, right, is the 4 water molecules, 4 oxygen molecules, right, in order to make water. So 24 divided by 4 is going to give you 6 O2. So this is why we know that you need the 6 O2 molecules. And this is, you also know this from your like exercise physiology. Labs, for example, is you've heard of the RER before, right? The respiratory exchange ratio, or your Q, your respiratory quotient. Well, what do, right? This equation is your volume of your VCO2 over your VO2. And you can do that math right here. How many carbons are there in this equation, right? There's 6. So you take your 6 CO2, that's the volume of carbon dioxide being expelled, over the volume of O2 that's being consumed, and it's 6. right? 6O2. And so therefore you get 1. So that's why if you burn carbohydrates, your respiratory quotient is 1, right? And now we can do the same idea if we're burning fatty acids, all right? So specifically palmitic acid. Now, again, this is a saturated fatty acid that we're talking about, and it's actually different. When you do beta oxidation with monounsaturated fatty or polyunsaturated fatty acids or odd-chain fatty acids, there are different steps in order to do all this. So we're just making it simple. We use palmitic acid, and like I said, we end up getting 7 FADH2s, 7 NADHs. We can add all of these up together in the mitochondria. Let's just write this equation here. We did this before. So we have C16 H32 plus O2. We're going to oxidize that with oxygen. And then we're going to make, you have 16 CO2 plus H2O. Actually, with this H2O here, how many water molecules do we have to make, right? If we're making 6 O2s, the reaction is right here, right? O2 plus 4 hydrogen ions making one water molecule. So you'd actually end up making 12 water molecules. Well, so to balance the equation, we could add plus 6 H2O right here. But then, of course, you could just balance it out and move the 6 and put 6 over here. That's why most textbooks have just the 6 water molecules. But you're actually making 6 water molecules through that oxygen step right there. So same thing when we calculate all the NADHs and FADH2s, right? What do we get? We get, if we put all of those together, we end up getting 46 of your NADH and FADH2 pairs, right? And that's going to be a combination of beta oxidation and the TCA cycle. Each one of those, right, has with it 2 electrons, right? So how many electrons are on 46 of these pairs? Be 92 electrons, right? So there's 92 electrons. And then how many electrons are required? We look back at the stoichiometry over here. We need an oxygen plus 4 hydrogen ions plus 4 electrons to make a water molecule. So we are going to need, we do the same thing. We divide that by 4, right? So we take 92, divide that by 4, and what do you get? You get 23. Yeah, 46. Yeah, 92. So you get 23 O2 molecules. So we can put that right here. That's how we know that this is consuming, you have to consume 23 oxygen molecules in order to completely combust this. And so this right here would also be, you could put plus 46 water molecules here, and then to balance that out, you could add, plus 30 H2O molecules, but of course you could subtract 30 from there, and you could put 16 over here, and that's again why most textbooks are going to have that. And now you could do the same idea, right? If you do the RER, right? So you have the respiratory exchange ratio, and you can do that same math. So you put your CO2 right here. So 16 CO2 over your 23 O2. And what do you get? About 0.7, right? And that's why when you do the gas analysis, your typical resting energy expenditure, or just at rest, what is the typical RER value, right? Around 0.8 or something, right? Because it's mainly going to be fatty acids that you're oxidizing, and it's going to be right within that range. And so this gives you a direct clue as to why it is that when you oxidize fatty acids, your respiratory quotient is so low. Now, the hydrogen ions, right? Here we get 32, we have 32 hydrogens that are being metabolized, because this whole thing only ends up going to 16 CO2, right? So you get 32 hydrogen ions, but look at this, 46 water molecules. 46 water molecules is how many hydrogen ions? Well, it's the same thing as electrons, right? 46 water molecules ends up being 92 hydrogen ions. So 92 hydrogen ions are actually being consumed to make metabolic water? Well, again, there's only 32 hydrogens here, and a lot of these are just being provided by, or not being provided as hydrogen ions at all. We can go through the actual steps to see where exactly hydrogen ions are being produced at, but it doesn't really matter because a lot of them are being just dehydrated in their water molecules. But the point is, the oxidation of a fatty acid ends up becoming net alkalizing, essentially. And now what we can also do then is look at what's known as the phosphate-oxygen ratio. Okay, so if we look at for fatty acids and for glucose. So here we have glucose. Well, we just said we made about 32 ATP. Let's say that you're in your muscle cell, and so you're making 30 ATP, okay? 30 ATP, so you have 30 of your phosphates, right? And how much oxygen do you need to make 30 ATP? You need 6, okay? So 30 divided by 6 is what? 5. Okay, so you have a 5 phosphate to oxygen ratio. If we said 32, then you'd be 32 divided by 6. So now you're like 5.3 phosphate oxygen ratio. Okay, now we do the same thing with beta oxidation. How many ATP? Okay, we can count this out. In fact, we have the NADHs. Remember, there's 2.5 ATP per NADH and then 1.5 ATP for FADH2. So you end up getting about 108 ATP in total. But remember this step right here, we basically use 2 ATP, so minus 2 ATP right here, and you get 106 ATP. And now we can do that calculation. So we get 106 ATP. We divide that by the total amount of oxygen that we need, so 23. So 23, and divided by 23 oxygen molecules equals 4.6, okay, phosphate oxygen ratio. What does this tell us? It tells us that, okay, start here. For every, you get 5.3 phosphate or ATP per oxygen consumed when you metabolize glucose. So your phosphate to oxygen ratio is 5.3. So you get 5.3 ATP per oxygen molecule. Here, it's 4.6 phosphate to oxygen. So look, you actually get more ATP with your oxygen. Per oxygen, you get more ATP per oxygen using glucose than you do using fatty acids. This is the reason why. But then we think, but I thought fatty acids provide you more ATP because more energy. Well, yeah, because you have, right, there's 16 carbons, C16H32, it's a big molecule, and so you end up getting 106 ATP. But when you do on a phosphate to oxygen ratio, this is what you get. 5.3 phosphate to oxygen, 4.6 phosphate to oxygen. So this also explains why if you go like a higher elevation, hiking a mountain or whatever, that higher elevation, initially you end up doing more glycolysis because you can make more ATP as well per oxygen molecule. So it's more efficient in that way. You're able to get more ATP per oxygen molecule than you do with fatty acids. Even though we tend to think that fatty acids is that's an oxidative phosphorylation. It is, but remember, we're burning glucose in the cytosol, but then the whole process of oxidation occurs in the mitochondria through pyruvate and then to acetyl-CoA, right? So if we're using oxygen to burn the entire glucose molecule, then this is why you get more ATP from per glucose molecule, the phosphate oxygen ratio, than you do with the fatty acid. All right. So that's another important aspect of using our energy substrates and what they actually mean. So one more important thing, which is interesting because, as I mentioned, when you're going up to higher elevation, glucose becomes a good fuel. Oh, and then the one more thing we're talking about, like the higher elevation, is you have the higher phosphate oxygen ratio. When you do glycolysis and you're making 1,3-bisphosphoglycerate, remember there's a lot of other pathways that are coming off. All these intermediates, pretty much you can make another one. Like we talked about with phosphofructokinase 1, there's the other enzyme over here, phosphofructokinase 2, that's converting this to fructose 2,6-bisphosphate, and that allosterically regulates the PFK1. You have a lot of things that you make in xylulose phosphate and other things, but this one here 1,3-bisphosphoglycerate, you can make this other molecule 2,3-bisphosphoglycerate. And this molecule actually influences the conformational shape of the hemoglobin and changes it so that it releases oxygen it releases oxygen easier. So you're able to use the oxygen better when you are at, say, higher elevations. So as you go at higher elevations, all of a sudden you get more 2,3-bisphosphoglycerate and you're able to get oxygen so you can use that. So glycolysis is really an amazing pathway because of so many things that it regulates and the feeder pathways that it does. And we couple that with oxidative phosphorylation, and now you're able to burn energy over and over again, and you don't have to worry about it's less likely you're not going to be acidifying yourself. I just want to say, because when you do the math right here, we can see that it makes sense that when you're doing a long, slow exercise, you're not breaking down ATP near as fast, and you have kind of a net alkalizing effect of oxidizing those fatty acids. And that could potentially be another reason why you can continue going longer with that exercise. Although, as I had said before, the idea that hydrogen ions and the acidity is the direct cause of fatigue is really not accurate. It's not supported by the research. It's really the combination of the phosphates, the hydrogen ions, the binding of the calcium, these metabolic waste buildups that's going on. But it's just it's an interesting aspect to also consider the influence of oxidative phosphorylation and what that does to the hydrogen ions. So hopefully this was helpful, and we can go through our next pathways next time.